In this video, we're going to talk about the important nucleotide containing the compounds in metabolic pathways. In the last topic, we talked about the metabolism and we already learned that there are two metabolic reactions. The catabolism, which is the breakdown of the large molecules into smaller molecules, and the anabolism, which is the small biochemical molecules are joined to form larger ones. Now, in this video, we're going to focus on the high-energy phosphate compounds. High-energy phosphate compounds is also known as energy-rich compounds. It is a phosphate linkage present in a certain intermediates of carbohydrate metabolism and containing the energy used in metabolic processes or transferred or stored. This is the structure of the phosphate. As you can see, there are three negative charge. High-energy phosphate are essential in a very important molecule we call the ATP. Here we have the structure of the adenosine triphosphate. Notice that the ATP usually has three phosphate connected with several phosphoanhydride bands. And there's a ribose sugar and the structure of the adenine. So, this is the entire structure is what we call the ATP. Now, if I were to use water to hydrolyze this first phosphate here and break this bond, this would chop the phosphate off and leave me with the rest of the molecule which we call the ADP or the adenosine diphosphate. Likewise, if I were going to use another molecule to chop the second phosphate to give me now what we call the AMP or the adenosine monophosphate. And lastly, if I were to chop off this last one here and just leave me which over here to the right, this would now give me the adenosine. So each of these phosphate here has a particular name associated with it. The first phosphate that is close to the sugar is what we call the alpha phosphate. The middle one is the beta phosphate and the terminal one is the gamma phosphate. High-energy phosphate compounds have greater free energy of hydrolysis than typical compound. They contain at least one reactive bond in what we call strained bonds. Energy to break these bonds is less than a normal bond, or the hydrolysis of high-energy compounds give more energy than the normal compounds. The more negative the free energy of the hydrolysis, the greater the bond strain. In chemistry, we talk about the energy that is produced in a reaction. We typically are going to use the unit of what we call kilocalorie or the calorie. Typically, the free energy release is greater than 6.0 kilocalorie per molecule indicative of the bond strain. Strain bonds are represented by a squiggle bond. High-energy phosphate groups are mainly classified into 5 groups. The first one is the pyrophosphates, the second one is the enol phosphates, the third one is the acyl phosphate, and the fourth one is the thiol phosphate, and lastly, the guanidophosphate. So, pyrophosphate has an energy bond is what we call the acid anhydride bonds. It is formed by the condensation of the acid group of it or its derivative. An example of it is the ATP. And then the second one is the enol phosphate, which has an enol phosphate bonds, formed when phosphate group attached in the hydroxyl group, which is bonded to a carbon having a double bond. An example of this is the phosphoenol pyruvate. And then the acyl phosphate formed by the reaction between the carboxylic group and the phosphate group. The example of it is the bisphospholiglycerate. And then the fourth one is a thiol phosphate. In this type of phosphate, high-energy phosphate bond is absent. Instead, high thioester bond is present. It results from the reaction of the thiol and the carboxylic group. An example of this is the acetyl-CoA. And lastly, the guanidophosphate is also known as phosphagens, has a guanidine phosphate bonds. It is formed by the attachment of the phosphate group to a guanidine group. And the most important compound in this bond is the phosphocreatine. And that's all for the high energy phosphate compounds.
So, good day, ma'am. So, my report for today, or my part of our report, is the adenosine phosphate, ATP, ADP, and AMP. So, let's tackle about the differences of these three phosphates. So, ATP, or the adenosine triphosphate, has 3 phosphate group, while ADP, or adenosine diphosphate, has 2 phosphate group. Meanwhile, AMP, or the adenosine monophosphate, has only 1 phosphate group. So as you can see in the picture, ATP has a nitrogenous base, which is the adenine, and ribose and 3 phosphate groups, while the ADP has a nitrogenous base that has 1 ribose and 2 phosphate group, while the AMP has adenine, ribose, and only 1 phosphate group. So let's tackle about the adenosine triphosphate. So in order for us to understand the ADP and AMP, we need, or we must understand the adenosine triphosphate. So adenosine triphosphate is a nucleotide consisting of an adenine base attached to a ribose sugar, which is attached to a 3-phosphate group. It is also the energy formed stored in cells. It is also obtained from the oxidation of wood. And also, it is the energy produced from these reactions is used for cellular reactions. So, this is, in this picture, this is the formula on how ATP converts to ADP, or how ADP converts to AMP, and ATP to AMP. So, in removing one phosphate group, it is called hydrolysis. So, in order for hydrolysis to occur, we must, or it needs a water, or H2O. So as you can see in the picture, ATP plus H2O converts to ADP. So inorganic phosphate plus hydrogen ion plus energy. So in this stage, ATP, one phosphate group was eliminated, so it has only 2 phosphate groups, and then that's the time you can call it ADP, or adenosine diphosphate, because it has only it now it's now only it has now 2 phosphate group. Yeah, sorry for that. And yeah, so ADP plus H2O converts to AMP. So like what I said earlier, ADP has only 2 phosphate groups, right? So in this stage, one phosphate group is removed. So now it has only 1 phosphate group. And then now you can call it AMP, or adenosine monophosphate. Yeah. So if in ATP, like it converts to directly to AMP. So here's the formula. So ATP plus 2H2O. From what I have studied, it is like 2 ADP. So like 2 ADP. So like you can say that the two phosphate groups were removed directly. So if two phosphate groups were removed directly, now it has only one phosphate group, and then we can call it AMP. Yeah, that's right. So when the phosphoanuride bonds are broken, input of energy is required. So the conversion value of ATP to ADP or ADP to AMP is negative 7.3 kilocalorie. Sorry for that. Negative 7.3 kilocalorie molecule. Yes, I forgot to put K. Sorry for that. So the conversion value of ATP to ADP is negative 7.3 kilocalorie molecule. Yeah, so like it is the energy that was used for the hydrolysis. Yeah, so now it can converts to ADP from ATP. Yes. So let's say when the cell has extra energy gained from breaking down food that has been consumed, it stores that energy by reattaching a free phosphate molecule to ADP, turning it back to ATP. Yes. So as what as what I have said, if like for example as humans we consume food, yeah. So that food will be like it stores energy. So after storing that energy, so like the free phosphate molecule which is from the food that we consumed, it will be reattached to the molecule or to the ADP. So if one phosphate was reattached to ADP, then it has now three phosphate groups, right? Yeah, so it comes back, it goes back to ATP. Yes, same as with the AMP. If there is free phosphate molecule reattached, then it goes back to ATP. Yes, so like I will give you an example. So like think of it as the energy currency of the cell. So let's say it is like a battery. Yes. If it is fully charged, then you can say it and you can say if it is fully charged, you can say it is an ATP. Yes, it is an ATP. But when it's running down, then you can say it is ADP. Yes. And when it is in a low battery state, then you can say it is an AMP. So like if so like you can relate it to a battery. Yes, I know you get it na. Yep. So I guess that's all for adenosine phosphate. I am Jindrik Chatilano. Thank you.
So there are also some important nucleotides containing compounds. So this is the flavin, adenine, and the nucleotides abbreviated in the FAD. So this is one of the major coenzymes in the metabolic reaction. So This is a requires numbers of metabolic reactions, redox reactions. So it is composed of flavin, ribitol, and that forms ribose, and the ADP, the adenosine diphosphate. So it has a nucleotide in it. So, and the basics things that it does a oxidations, reductions. It accepts and donates electrons. So here we have the FAD, this accepting two electrons. So gaining electrons is a reductions. It is a reductions. So it is forms of FADH2. So losing electrons is a oxidation. So we get the reductions and oxidations. And the FAD goes back and forth to the FADH2 as we needed providing electrons. Here's the actual structures of it. So we got the adenine, and there is a sugar. So two phosphorus, and the ribitol, and the flavin. So this type, so typical cell reactions which is the FAD serve as oxidizing agent. So involves conservation of alkyne to alkyne. In additional, so the FAD, or the flavin adenine dinucleotide, is accepts only two hydrogens atoms. So it is involved only in the Krebs cycle. So it is not involved in the other like glycolysis link reactions. It only involved in the Krebs cycle. So that's all.
Good day everyone. So my topic is about nicotinamide adenine dinucleotide. So I am going to explain first what is the nicotinamide adenine dinucleotide. So nicotinamide adenine dinucleotide is a coenzyme central to metabolism that found in all living cells. NAD, or nicotinamide adenine dinucleotide, is called a dinucleotide because it consists of two nucleotides joined through their phosphate groups. One nucleotide contains an adenine nucleobase and the other is the nicotinamide. NADH stands for nicotinamide adenine dinucleotide. NAD plus hydrogen. This chemical occurs naturally in the body and plays as a role in the chemical processes that generates energy. So the nicotinamide adenine dinucleotide consists of two nucleosides joined pyrophosphate. The nucleosides each contain a ribox ring, one with adenine attached to the first carbon atom in the one position, and the adenosine diphosphate ribose in the other with nicotinamide at this position. So this is the sample picture of the structure of nicotinamide adenine dinucleotide.
So this time, we will be discussing what is coenzyme A. So it is one of the important nucleotide containing compounds in metabolic pathways. It is a derivative of pantothenic acid, which is a vitamin B. So this one has a function not to oxidize and reduce but to transfer acetyl groups. The active form of coenzyme A is the sulfidyl group or the SH group, the thiol group. it changes between CoA form and its acetyl-CoA form. So, acetyl-CoA form is very important molecule in TCA cycle. Remember, the cells uses glucose and oxygen in order to create energy. So glucose have 6 carbon molecules. During glycolysis, we break these molecules into 2 pieces, each having 3 carbons, and that's what we call pyruvate. And when we enters the pyruvate dehydrogenase complex, where one of those carbons gets snapped off and eliminated as CO2, and what we're left with is a 2 carbon group known as an acetyl group. So why it is called acetyl group? So the most simple hydrocarbon molecules are called alkanes, and 2 carbon alkanes is called ethane. If we take the ethane and put a carboxyl group in it, it's now known as ethanoic acid, and the common name of it is acetic acid. If we take a molecule of acetic acid and turn it into a substituent, then we will have to use the suffix YL, and now it becomes a acetyl group. So basically, coenzyme A helps with the energy production within the body. It is a helper molecule, and it is also a non-protein chemical substance needed for activation of some enzymes, the proteins that catalyze or activate important chemical reaction with the body or within the body. So here we can see the structure of coenzyme A. Vitamin B5 is made of this. Basically, the CoA is made up of this molecule, which are the B5. These 2 amino ethanethiol, the pantotonic acid, and these 2 ADP, these 2 little pentagon here in the side, and the phosphate group, which means this part here can be attachable with another molecules. And then basically the most important thing here is the HS group, and that's all for the coenzyme A.
Now, we have the last part of the important nucleotide-containing compounds in metabolic pathways. Classification of metabolic intermediate compounds. So in classifying these metabolic intermediate compounds, these can be classified into 3 groups based on their functions. The ATPs, or the adenosine triphosphate, to ADP, or adenosine diphosphate, to adenosine monophosphate, or AMP, are classified as the intermediates for the storage of energy and transfer of phosphate groups. Now, the AFD, or the flavin adenine dinucleotide, and the NAD, or the nicotidamine adenine dinucleotide, are classified as the intermediates for the transfer of electrons in redox reaction. And the acetyl-CoA, or the acetyl coenzymes A, are classified as the intermediates for the transfer of acetyl groups. These metabolic intermediates can be classified as subcontractors. These are molecules that are precursors or approach preceding another in a metabolic pathway, or metabolites, or the end product of metabolism of biologically significant molecules. Although these intermediates do have only tiny direct impact of cellular functions, these compounds can play important role in allosteric regulation, or the regulation of enzyme by binding an effector molecule at the site of other enzyme site, or the site to which the effector binds in terms of alloretic site or regulatory sites of enzymes. And that is all for the important nucleotide-containing compounds in metabolic pathways. Thank you for listening.